If you're an epigenetics fan, you may have read that there are already assays out there for the 4-mil and the carboxyl. I have serious reservations about those assays, but we can talk about those if you want. We'll also help you with your ChIP-seq, whether it's for histone modifications or for the writers, the readers, and the erasers of histones and methylation marks. We can help you with those. There are different ways in we can help you. Mostly these assays will be for genome-wide distribution. I believe for the most part, people who want to do single locus ChIP in their labs, they're generally set up. But I've already taken some consultation with people who were not set up to do ChIP in their labs and wanted some advice. So we can help you with that as well. So, in more specific, what services do we offer? So for genome-wide DNA methylation, you're going to have a choice, and we're going to spend the bulk of this talk discussing what are the options for DNA methylation. You have your bisulfite-based methods, and that's whenever it says BS, that's bisulfite sequencing. So we have whole genome bisulfite sequencing. We have reduced representation bisulfite sequencing. If you think about it, cytosines, CpG sites, are not evenly distributed across the genome. And within those CpGs that are not evenly distributed, methylation is not evenly distributed. So if you have something that's asymmetrically distributed across the genome, it makes sometimes little sense to sequence the whole genome, because 70% of your reads will not have a single informative CpG. And that is what Alex Meisner was thinking when he developed RRBS, reduced representation bisulfite sequencing, in which using a restriction enzyme, he just uses that to reduce the genome to the parts of the genome where the CpG sites occur more frequently. And then we just do bisulfite sequencing. The ERBS is enhanced reduced representation bisulfite sequencing. That's a modified version of the same assay. We just did it in my previous lab because we thought that the RRBS was too CpG-centric. And since RRBS was developed in 2005, many papers came out that demonstrated that a lot of the informative CpGs were not sitting at CpG islands, but rather at regions flanking those CpG islands called CpG shores, and even further intergenic at enhancers and other regulatory regions, and now the new super enhancers. All of those have DNA methylation, and they're not at CpG islands. So by doing this modification, this enhanced reduced representation, we are capable of capturing more of those non-CpG island CpGs. So it's a good trade-off between doing the whole genome and paying for a lot that you don't need, and doing RRBS where you're only looking at CpG islands. So this is kind of a happy medium. Both of these methods are bisulfite sequencing based, and they're going to give you base pair resolution. The other option is to do methylated DNA IP, MEDIP. In this case, it's very much like a ChIP-seq assay where you use an antibody to enrich for methylated cytosines. Then you pull it down and you sequence it. And last but not least, we still have the Illumina Infinium arrays available. Those are a good option. These will offer you about 450,000 CpGs in the genome. They're usually a good resort for people who want to do very large cohorts of patients, epidemiological studies, for example, or profiling studies where you want to do hundreds of samples and you don't want to be paying the cost of next-gen sequencing for all of those. The limitation, they're only for human. And that's a question we get very often, both at the sequencing core and at the epigenomics core, is that these assays are not available for, these arrays are not available for mouse or rat or other model organisms. So that's the one limitation. So what if you want to study hydroxymethylation? Those of you who have been following the literature may be aware that there's at least 5 different assays out there that claim they can measure hydroxymethylation. Again, I have serious reservations with many of them, and I have a bill from the sequencing core to demonstrate that they don't actually work. So we have decided to restrict our hydroxymethylation to an antibody-based method. This is like ChIP-seq. It's robust. It always works. It won't give you base pair resolution, though. So that's a limitation, but it's there, and it gives you genome-wide hydroxymethylation. If you want to do genome-wide histone modifications, or the readers and the writers of the epigenome, we're still using ChIP-seq. This is the assay that you guys all have known, and many, if not most of you, have used over the last few years. What we will do for you is we'll help you with the library preparation. Most labs are very good at doing ChIP. Not everybody wants to do the library preparation themselves. I should say Bob's core also offers library preparation for the ChIP-seq. Both his core and our core will ask you for 5 to 10 nanograms of DNA. If you give us any less, it's at your own risk. But we'll do the library preparation for you. And as I said, this helps save on reagents. And also, you have someone who has extensive expertise in preparing these libraries, doing it for you rather than having to learn it from scratch. What we will very soon, not yet, but we're hoping that towards the end of the year we'll be able to offer, if not early next year, is something called N-ChIP, native ChIP. So anyone who's been doing chromatin IPs has probably been cross-linking the proteins to the DNA, sonicating, and then doing your enrichment test, correct? Now, native ChIP, that has one problem. Every time you change cell type, cell concentration, cross-linking conditions, tissue, even operator hands, your sonication conditions change, and they can take months to re-optimize or even to optimize the first time. So sonication is usually the first hurdle, particularly if you need to get very small sonication for ChIP-seq. Native ChIP doesn't use cross-linking. It uses micrococcal nuclease to bring down the chromatin to single nucleosomes or dinucleosomes. So now you have very small fractions of the genome at the single nucleosome level resolution. It's not dependent on cell type. It is dependent on cell number, but we have titrated it to different cell number conditions. And after that, so now that I don't have to optimize for each of your own cell types the sonication, now I can offer you chromatin IPs for validated antibodies. So that way, if you don't want to do the ChIP in your lab, you don't have the personnel, you don't have the bandwidth, you don't have the patience or whatever it is, we can do it for you, or we hope we will be able to do it for you very soon. This assay is working in our hands. We're just not ready to scale up to that extent at this point with the personnel we have. We will only offer that for validated histone antibodies. We will not be doing it for transcription factors. We will not be doing it for rare proteins because that would require a lot of optimization, and we cannot be custom optimizing assays for each user. So we feel that these are the assays we can offer robustly to the whole community. All of those were genome-wide genome-wide assays for the epigenome, but sometimes you're not interested in genome-wide. You may be interested in 1, 2, or a small panel of genes. Particularly in epidemiological studies, you may be interested in a few genes across thousands of samples. And that's where these two assays, mass array or pyrosequencing, become very good and actually have a very good cost-benefit, because you can get many, many, many samples with just a few primers, and then the cost per sample becomes quite low. If you want to do the opposite, which is what I generally do, which is many primers and a few samples, it's not as cheap, but it's still a very useful assay because it will give you quantitative base pair resolution of long stretches of DNA. So with mass array, you can do up to 400 or 450 base pairs. So with 3 or 4 primer pairs, you can span a whole promoter and capture almost every CPG in that promoter. So now you have very comprehensive study of one gene or a set of genes. At a very quantitative, this is considered the gold standard in the field these days. Either of these two assays are considered the gold standard. And finally, I already mentioned this, we'll offer bioinformatics services for epigenomic data sets. We'll give you all the data that we generate in any of these assays. We will not give it to you until we are satisfied that it has passed all our quality controls. And if we feel that the data is not up to the quality control it should be, we will repeat the assay at our own cost. So, because we believe it's something we did wrong. As a fee-for-service, though, we will offer data processing, biological comparisons, and even integration with either your own data sets that you may have from previous experiments or publicly available data sets like ENCODE and TCGA. So all of those things will be offered as service through the bioinformatics epigenomics core. So before we go into how should you choose your assay, for those of you who are not familiar with bisulfite sequencing or even bisulfite conversion, I did want to go over this briefly. So the reason why we use bisulfite as a reagent in epigenetics is that when we expose cytosines to sodium bisulfite, an unmethylated cytosine will go through a deamination and desulfonation reaction until it converts into a uracil. So it loses the amino group, becomes a uracil. Methylated cytosines, and even hydroxymethylated cytosines, are resistant to this deamination process. So by taking genomic DNA and exposing it to sodium bisulfite, we will have this differential behavior of the methylated and the unmethylated cytosines. So if we represent it here, you have your methylated DNA, you expose it to bisulfite conversion, nothing happens, you go through a PCR reaction, and your Cs that were initially Cs are still Cs. However, in the unmethylated alleles, what's going to happen is that your CpGs deaminate to uracil. During the PCR reaction, this uracil is replaced with a thymine. So now what we created is a C to T SNP that can be detected in different ways. We can use restriction enzymes, so we've been using that for many years. We can use, we can differentially amplify this using different primer pairs that either recognize the CGs or the TGs. Or what we do in the core, we can either differentially hybridize it onto a C to T SNP array, that's the Infinium array, or we can sequence it, and we will find that C to T SNP in our Illumina sequencing results. However, as I said, hydroxymethylation does not deaminate. So now we have a problem. We have methyl cytosine that doesn't deaminate, but we also have hydroxymethyl cytosine that doesn't deaminate. The problem with that is that HMC is present in varying quantities in different tissues. It's very high in brain, it's very high in ES cells. So if you work with brain or ES cells, you certainly cannot ignore this. If you're going to do a bisulfite sequencing reaction, you need to somehow measure in parallel the hydroxymethylation. Otherwise, you will misinterpret some of the Cs as methylated when they're actually hydroxymethylated. The other issue is what happens in tissues where it's low is that it becomes very difficult to detect. And that's why many of those assays that I said are out there are actually not reliable, because they're very noisy. And it's very hard to detect these very low quantities of hydroxymethylation. And that's where the antibody actually performs really well. However, even in tissues where hydroxymethylation is very low, but even more so in tissues where it's high, we can't just ignore it. Because the big surprise that came to all of us when we were studying this is that this is a genome-wide correlation of gene expression changes and DNA methylation changes. And when I first started my postdoc back only 3 years ago, when I first started my postdoc, my mentor sat down with me with a notebook, a notepad, and a pen, and he drew this beautifully inverse correlated genome-wide study that this was going to be figure 1 in my paper, where all the genes were going to be inversely correlated, their DNA methylation and their gene expression. Guess what? I never published that figure, neither did anyone else, because the truth is, the elephant in the room in methylation for many years was that on a genome-wide level, not all genes are strongly correlated or inversely correlated with their methylation status. Some genes, where they had done the initial studies, are certainly regulated through promoter methylation, but this is not true genome-wide. What we found, though, was that when we looked at the same comparison, now gene expression against hydroxymethylation, the R value now became 0.75. So we went from a very weak inverse correlation for methylcytosine to a very strong positive correlation with hydroxymethylation. So even though the mark is far less frequent than the cytosine methylation, it's that much more stronger in determining transcriptional regulation. So we can't just ignore it. Hydroxymethylation is here and is here to stay, and we have to measure it when we're thinking about the epigenome. So you've decided you want to do epigenetics, and I just told you there are all these different assays, and you don't know which one to choose. Well, it's not just a matter of taking the menu and saying, I like this one. You're going to have to decide and take many things into consideration. Your first thing would be, well, do you need genome-wide, or do you only need a few or a single locus? So there's no point in doing genome-wide if you're only interested in a few, because we have very good assays for restricted loci. The other question is, do you need base pair resolution? Are you interested in specific CpGs, or will a regional information as given kind of like a ChIP-seq or an AMIDIP experiment be enough for you? Are you interested solely in methylcytosine? Do you also want hydroxymethylcytosine? My advice would be that you want, but you may choose not to. Do you also care about histones? Do you have good or bad quality DNA? Is your DNA high molecular weight extracted DNA, or is it, paraffin-embedded DNA that's usually lower quality, or, God forbid, trisol extracted DNA that is even worse? Do you have cell lines and you have abundance, or do you have sorted or laser microcapture dissected DNA tissue? That's going to restrict the amount of cells, it's going to restrict the assays you can do. And finally, do you have cost restrictions, or are you lucky enough that you don't? So the first two considerations, the first choice you're going to need to make is, do I want to go bisulfite-based, or do I want to go affinity-based or antibody-based? And there are pros and cons. So bisulfite-based methods have base pair resolution, and they can offer precise quantification, very precise. They'll tell you 73% methylated. However, they can have false positives due to incomplete conversion. That's the #1 cause if you're not savvy at doing the bisulfite treatment. It will not distinguish hydroxymethylation, so it will give you a false positive there. It requires very high quality DNA. So if you're extracting your DNA from histological slides, like some people do and other things, you may not be able. There are tricks around it, and we can work with you, but it's not going to be the best quality assay. The bisulfite DNA is unstable and cannot be stored. So once you do it, once you decide to do it, you have to do it. You can't just stop halfway and maybe next month when we get the grant to do it. You do it now. And finally, when all of these bisulfite-based methods, whether they're genome-wide or single base or single locus, they require a PCR step. And this PCR has to be very well optimized to amplify in equal efficiency the methylated and the unmethylated representation, because the minute you don't amplify those with equal efficiency, you bias your results. And the #1 reason why people I've sent out the protocol to fails is that they change the tack, because it's a very expensive tack. But I don't know, they may think I'm stupid and I like to pay more for a tack. It's not. It's that that's the tack you need. So you can't just change it. So this is a very, very crucial step. Affinity-based methods, on the other hand, they can be performed with lower quality DNA. They do not depend on bisulfite treatment. If you have good antibodies, and we believe we do have good antibodies, they should not cross-react with the other modifications in cytosine. And you get to query the whole genome without sequencing the whole genome. So even though you lose your base pair resolution, you still have your whole genome without sequencing 70% of the genome that doesn't have base pairs, doesn't have CPGs. Of course, it's not going to be base pair resolution. It can be dependent on CPG content, so areas that are poor in CPG are harder to pull down. We have very good optimized protocol, but still, we're not going to get every single region. So when there's lack of signal, you won't know if it's totally because of lack of methylation or because you just can't pull down there. So you will need some form of validation for those lack of signal. And of course, like all antibody-based methods, they're not the best signal-to-noise ratio, but we can do several things in the protocol to optimize it. So finally, you decided you want to do a bisulfite-based genome-wide method. So actually, not genome-wide, because I added a column there. So you're doing DNA methylation. And this would be kind of like your cheat sheet. If you want to do DNA methylation, these are all the questions you should ask yourself, and these are all the assays you have, and that's based on what you answer, yes or no. This is the assay you will ask. So if you want to do a few loci, you're going to go with mass array or pyrosequencing, and it's going to be quantitative and base pair resolution. We require about 10 nanograms of DNA per amplicon, and the cost is per sample varies on whether you're going to do few primers, many samples, or few samples, many primers. So it's hard to give it a dollar amount, but we can help you with that. And then if you do your bisulfite sequencing, it comes in 3 flavors: whole genome, ERBS, or the newer version, multiplexed ERBS. So this one will give you the whole genome with a lot of wasted sequences. These two will give you about 3 to 4 million CPGs. So you're about 20% of the autosomal CPGs are captured. They're all quantitative. They're all base pair resolution. And this is where the big difference is. You want to do whole genome and you want to get a decent coverage of 10 or I would say 30x, you're going to need about 3 lanes. Maybe 2 with the new technology, but we're not very certain, so I would still say 3. If you do ERBS, you're going to do 1 lane. And if you're going to do multiplex ERBS, anywhere between a quarter or half a lane, depending on how much depth you want. So why do ERBS instead of multiplex? Why not always do multiplex? Because the difference here, you pay it in DNA. We can do ERBS with as little as 2.5 nanograms of DNA. So now you're talking sorted cells, you're talking laser capture microdissected tissue, you're talking very, very pure populations. This one, unfortunately, we still require 75 nanograms, which is very good. When we started doing DNA methylation, my first protocol required 11 micrograms. So we've gone down a long way. But still, you won't be able to do it in very, very rare populations. But of course, if you can get 75, this will be cheaper. And finally, you can do MEDIP, that's the antibody-based. It'll give you whole genome. It's semi-quantitative, like a chip. By the intensity of the peak, you'll know the amount of signal. But it's not base pair resolution. Depending on the depth, you will require 1 to 2 lanes, and it requires more DNA. If your sample is very hypomethylated, you're going to have to give me as much as 1 microgram. If your sample has normal methylation, we can probably go down to 200 or even 100 nanograms. So all of those things need to be taken into consideration. If you treated your tissue with azacitidine, now you're going to have very little methylation left. We're going to have to compensate at this end in the amount of DNA you give us and things like that. So this is what would happen if you wanted to use the core. The first thing we would always require is that we have a consultation, even if it's a brief one, particularly so that we can know what to expect, but also so that we can help you and make sure that you're choosing what's right for you. You would then request the service through iLabs. We are now live on iLabs, and we already had our first submission through iLabs. So you can submit your samples, request the service through iLabs and submit the samples to the core. Then the core will make sure that we process your samples in the right assay and the timely manner. We would then submit the process sample to the sequencing core because we work very closely. In fact, we have regular joint meetings with Bob's core, so we function almost as a unit. Then they will give us the data back. We will perform all the quality control and any data analysis if that is what you requested. And then we will give you back the either raw or processed data to your lab. So if you want all these things, you're going to need to know where to find us. And this is where you find us. You find us at MSRB 22568. So this is the same place where you are dropping off samples for Bob's core. So it's the same room. We just have a separate section there. Claudia is there every day from 8:30 to 4:30. So don't try to submit samples at 6:30. It's not going to happen. And if you want more information, obviously you can call me or Claudia, but this is our URL for the Epigenomics core. And I don't know, I ended up going longer than I thought, but if you have questions, please, I'll be happy to take them.
Assays, that there were a number of core assays, and you made a comment about the DNA sequencing core's bill. We don't provide those assays.
Oh, no, that was my own.
And I hope you were happy with the sequencing.
No, the sequencing was great. It was the kits that were bad. No, sorry about that. No, I apologize for that. It was us trying to set up the assays, and unfortunately, the only way we have of finding out if it worked or not is to actually sequence it. The data was beautiful, but there were no enrichments. So, yes?
Do you have any statistics of turnaround time from when we first talk to you to when we get our data back? How long is that process?
This is Bob's favorite part, because I say all of these assays our core is very artisan-like. We have to manually process all of these things. We don't use robotics for any of these. So we have a capacity of processing about 10 samples. Some of these assays take a whole week. So whole genome bisulfite, all the bisulfite-based methods take a whole week. So we can do about 10 samples a week if you want whole genome bisulfite. If you want hydroxymethylation, we can probably do about twice that number in a week, maybe a little bit more. Then we send it to the core. There is the sequencing core queue, which is usually about 2 or 3 weeks. Sometimes if there's been issues with Illumina, a little bit longer, but usually about 3 weeks we get the data back. And then then it will depend on how much data analysis you want. If you want the raw data, you can have it back once we've done the QC, so in another couple of days. But if you want data analysis, that will depend on the complexity of the project you want. And of course, we are currently processing samples as we get them because we don't have much of a queue, and we're hoping that if we get to that point, we'll just increase the core and we won't ever have a long queue. That's what they promised me. Yes?
Why don't you compare 5MC and 5HMC pattern of Figueroa? Can we do a targeted analysis?
We can do hydroxymethylation at single locus by doing the same thing you would do with a chip. So we can do the pull down for you because we have it optimized, and then you would have to design target-specific primers. In fact, one thing I didn't say in all these steps is that we have many internal quality control steps in all our libraries. So whether you're doing single locus, you will need to test enrichment. But if you're doing genome-wide, we do our pull down, we test enrichment, we then prepare the library. This is also true for ChIP-seq. We test enrichment again. If we lost enrichment, we do not sequence that library. So one thing we would, even if we wanted to do single locus or genome-wide, we would always request that you give us target-specific loci where we can check the enrichment of your, in fact, if you give us DNA from a pull-down, we expect that you have tested your enrichment already. You're not going to prepare a library from something that doesn't have enrichment. So yes, we can do it similar to a chip experiment. Any other questions? No? Okay. Sure, this always happens. Is there any more questions? No.